Hello. We're continuing with cell biology and life science in a nutshell. We're learning about cell membranes. In a bit of detail. So the phospholipid bilayer, which consists of proteins. The proteins transport various molecules, large and polar molecules across them. They also serve as receptors. Um, which helps cells receive signals, um, causing various changes inside the cell. Also, some of them just serve for the purpose of connecting cells together, causing creating tissues out of multiple cells. Um, so just some notes that we had mammalian plasma membranes are more complex than, for example, E. coli membranes, which are just unicellular. Um, the major phospholipids in mammals membranes are phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, sphingomyelin, consisting of 50 to 60% of the total membrane lipid. In addition to phospholipids, the membrane consists of glycolipids, which are uh, carbohydrates that link up with the lipids, and cholesterol molecules as well, which help with the rigidity and the structural integrity of the membrane. Individual molecules, both lipids and proteins, are free to rotate and move in lateral directions. Fluidity is a critical property of membranes, is determined by temperature and lipid composition. Example, interactions between shorter fatty acid chains are weaker than those between longer chains. Membranes containing shorter fatty acid chains are less rigid than those with longer chains. So these ones that are less, um, they are more fluid at lower temperatures. So if it's colder, they're not as rigid because the linking is not as strong between fatty acid chains. Lipids containing unsaturated fatty acids similarly increase membrane fluidity because these have double bonds, these fatty acid chains, introducing kinks in the fatty acid chain. So the kinks cause more fluidity because the rigidity is not as strong in the membrane, and the packing is more difficult of the molecules. Um, because of its hydrocarbon ring structure, cholesterol plays a distinct role in determining membrane fluidity. Cholesterol molecules insert into the bilayer with their polar hydroxyl groups close to the hydrophilic heads of the phospholipid molecules of the bilayer. So it's here showing polar hydroxyl group of cholesterol molecule, which is in black and white, and the other molecules are the phospholipids in their bilayer structure. The rigid hydrocarbon rings interact with the region of the fatty acid chains that are adjacent to the phospholipid. So the interaction decreases the mobility of the outer portions of the fatty acid chains, making this part of the membrane more rigid. So since the polar ends are not able to interact as well, and the fluidity is decreased and rigidity is increased of the membrane. On the other hand, insertion of cholesterol interferes with interactions between fatty acid chains, thereby maintaining membrane fluidity at lower temperatures. So because the fatty acid chains cannot interact as well, um, the rigidity is decreased and fluidity is increased at colder temperatures. Proteins are the other major constituent of cell membranes. As we noted earlier, 25 to 75% of membranes are proteins. This is a model first proposed by Jonathan Singer and Garth Nicholson in 1972, viewing membranes as a fluid mosaic in which proteins are inserted into the bilayer. Proteins are anchored in the membrane by lipids covalently attached to the polypeptide chain. Proteins can be anchored to the extracellular face of the plasma membrane by glycolipids, which bind to lipids, and the cytosolic phase by fatty acids, or prenyl groups. So shown some examples of proteins. These are carbohydrates, membrane spanning alpha helix, fatty acid prenyl groups, integral membrane proteins, peripheral membrane proteins. Is this bright fuchsia one binding with the integral membrane protein, the purple. So some proteins go all the way across membranes. Those are integral. Peripheral are the ones that bind to the integral. They're on the like the periphery of the membrane. They can either be on the cytosolic side, inside the cell, or outside the cell, the extracellular side. The hydrophobic side chains of these amino acids interact with the fatty acid chains of the membrane lipids. Formation of an alpha helix neutralizes the polar character of the peptide bonds. Okay, so basically hydrophobic parts interact with hydrophobic parts, and hydrophilic parts interact with hydrophilic parts. And that's how the molecules interact. While phospholipids provide the basic structural organization of membranes, membrane proteins carry the specific functions of the membranes. So there's two classes, again, integral proteins embedded within the bilayer. Peripheral proteins are not inserted, but are associated with the membrane indirectly, generally by interactions with integral membrane proteins, as we saw right there. Many integral proteins, called transmembrane proteins, span the bilayer, so portions are exposed on both sides of the membrane. The membrane-spanning portions of these proteins are usually alpha helical regions of 20 to 25 nonpolar amino acids. So within the layer are the nonpolar parts, and exposed outside on either side of the bilayer are the polar sides of the proteins. So some transmembrane proteins span the membrane only once, others have multiple spanning regions. Most transmembrane proteins of eukaryotic plasma membranes have been modified by addition of carbohydrates exposed on the surface of the cell and participating in cell-cell interactions. Those are called glycolipids if they're binding to lipids, or glycoproteins if they're binding to proteins. Thank you.